now gonna be our next session. I think two doctors have like special, definitely here have special chemistry because I can tell like Dr. Alberto is smiling, and then Dr. Lee when Dr. Lee is having the speech. So I'll leave time to you, two doctors.
Okay, yeah, let me start. Okay, Alberto, I don't know. Okay, one question actually. Yeah, we are working same place, in Sweden. So I spent 1.5 years in Lund, and then I moved to Stockholm. So we in 1996 or 97. So question is that how many clinical trials have been done already for the horse antivenom, those antibodies? I just care about the safety, basically used for the human beings.
The antivenoms produced by Instituto Clodomiro Picado have been tested in 4 clinical trials in Colombia, 1 big clinical trial in Nigeria, and 1 clinical trial in Papua New Guinea, and 1 clinical trial in Sri Lanka. Basically,
no
severe
side effect, right?
Well, these are the clinical trials performed with our products, but of course, many more clinical trials have been conducted with antivenoms produced by other institutes. And essentially, the adverse reactions depend, of course, on are very closely related to the dose. Patients which have more severe envenomings receive a higher amount of equine antibodies and are more likely to have adverse reactions. In the case of the anti-SARS-CoV-2 antibodies that we are proposing as treatment for COVID-19 patients, they are going to receive a dose which is much smaller than the dose that receive snake-bitten patients having moderate envenomings. So we don't expect them to have severe adverse reactions at all, because the dose of equine antibody they are going to receive is very low in comparison with the one that is used to treat snake-bitten patients.
Okay, thank you. Is there any comparison between the horse serum and the human being serum that's from COVID-19 recovered patient? Is there any comparison? Is your cell testing?
Yes, the horse antibodies are about 80 times more potent neutralizing the virus in vitro.
That's really good. Of course, you purify, right? It's not like human beings serum directly used.
Yeah, no, but we have also been working in purifying human antibodies from convalescent plasma,
okay.
And the amount of neutralizing antibodies in the plasma of the horses is much higher, because of course these animals have been hyperimmunized. They have received 4 injections of viral proteins. So its immune system is really trained to produce antibodies against these viral proteins.
And also you immunize the horse with 2 different type of protein. Actually, one is single S1 protein, and another one is use mixture of protein to immunize the horse. Is there any difference between the serum neutralizing activity?
Not in vitro. In vitro, we saw the efficacy, or the neutralizing efficacy of both preparations is very similar. We are intriguing about the response in the clinical trials, because you know, in the neutralization test, we are measuring the capacity of the antibodies to inhibit the virus to enter into the culture cells. But in vivo, in the clinical trials, there will be another mechanism like antibody-dependent cytotoxicity of other mechanisms that could be involved and could improve the response of the patients or could cause adverse effects. We don't know. So, but in vitro, we didn't see major difference between both preparations.
Dr. Alape, why don't you share with Dr. Lyu also the work that you're doing with the other protein now that is trying to be developed that has an equine tag and that may allow for greater potency of what is produced?
Yes, at this stage, we are trying to optimize the yields of the production line. And one of the strategies that we are using is to express the viral proteins as fusion proteins with an FC portion of the equine IgG4 to because this has been proven to enhance the response of the animals. If you immunize them with a protein fuse with the FC tag of the IgG, it will be the immune response will be better. So at this point, we are trying to increase the yields and reduce the cost of the process to have a product which a better cost effectiveness relationship.